Hi, my name is Hyun Kim, and I'm from Korea Advanced Institute of Science and Technology. Today, I'd like to talk about our recent work on TCR-epitope interaction prediction. TCR-epitope binding is crucial to the adaptive immune system activation, and many TCR-epitope binding prediction methods based on machine learning have been published over the past few years. However, prediction is still difficult for unseen epitopes, which are epitopes not seen during training. And the reason for this is the scarcity of currently available data. Here we propose a model called TSpred, short for T-cell receptor binding specificity predictor, which is trained on paired chain TCR data. The datasets we use in this study are five-fold cross-validation datasets, and we use two different splitting strategies to divide the dataset into 5 partitions. One is random split, which is simply randomly splitting all the pairs in the dataset. And the other is strict split, where the pairs are divided so that the peptides are non-overlapping across the partitions. So random split leads to seen epitope prediction, whereas the strict split leads to unseen epitope prediction. There are two commonly used strategies for generating negative data, which we also use in our study. The first is random shuffling, RS in short, which is shuffling the positive TCR peptide pairs in the dataset to generate negative pairs. The second is sampling from negative control data, NC, and this is sampling from the TCR repertoire from healthy donors without known epitope specificity. The problem with this approach is that the positive and negative samples have TCRs drawn from different background distributions, which can make it too easy to distinguish between positive and negative pairs. So while both strategies have limitations, the random shuffling strategy is generally preferred. These are the data sets that we use. And the first two data sets, NetTCR and IMREP, are obtained from previous publications and belong to the seen epitope prediction task. On the other hand, strict NC and strict RS are newly created based on the NetTCR dataset and belong to the unseen epitope prediction task. As the name implies, strict NC is constructed with the negative control sampling strategy, whereas strict RS is constructed with the random shuffling strategy. The model architecture is an ensemble model of a CNN-based model and an attention-based model. Each model takes as input the peptide and 6 CDRs and outputs the binding probability. In particular, the reciprocal attention component of the attention-based model was designed specifically to extract the patterns underlying TCR-epitope interactions. These are the results on the seen epitope prediction. So we compared our method with 5 other state-of-the-art methods, and TSpred achieved the highest mean RCAUC on both datasets. It achieved 0.86 and 0.8 on NetTCR and IMREP respectively. And these are the results on the unseen epitope prediction task. We could observe that compared to the seen epitope prediction, models perform generally much worse on the unseen epitope prediction. Still, TSpred achieved the highest mean RCAUC on both datasets, achieving 0.65 and 0.55 on strict NC and strict RS, respectively. We also analyzed attention scores from the reciprocal attention mechanism using structural data. So using 24 structures from SDCR-DAB, we compared the attention scores of the close and far peptide CDR beta residue pairs. And we were able to observe a significant difference between the two score distributions. In summary, TSpred shows state-of-the-art performances on the seen and unseen epitope prediction tasks. And it also offers model interpretability by capturing the structurally close TCR peptide residue pairs. Thank you for your attention. These are my acknowledgements for my supervisor, collaborators, and lab members. The preprint is available online, and the manuscript has been recently accepted to bioinformatics. I'm also looking for postdoc positions, so please see my CV if you're interested. Thank you.